All right, thank you, Sandra, for this amazing introduction. Thank you all for being here today. I promise to deliver my dissertation work in the simplest way as I can. So if there is a slide or two where you think things are getting more complicated, please bear with me. I will try my best to summarize it in the simplest way. And vice versa, if you think things are super easy for you, please have no mercy on me. Ask me really hard question, or deep question, not hard, deep question, during the question time at the end of this talk. So my dissertation entitled Unraveling the Function and Regulation of circRNA's in Aging and Age-Related Disease. So let's start. Aging. We all are experiencing aging as we're getting older, right? And it did not go, so aging is a time-related drop of physiological function that are survival for are important for survival and fertility. It's associated with so many changes as biological changes, physical changes, and so on. Now, how many times have we went to a doctor with our grandparents or grandparents and wonder why they are sick or what makes them ill? And usually we hear something like, it's very common for their age, or that's because they are getting older. So that's actually true, because aging is a risk factor for so many diseases, including cancer, heart disease, and Alzheimer's disease. During my research, I wanted to focus on aging and Alzheimer's disease in particular, and let me tell you why I wanted to study Alzheimer's disease. So here are some facts about Alzheimer's disease. Sadly, it's the 7th leading cause of death among all ages in the United States, based on a recent data by the CDC. If you notice with me here, it used to be the 6th one, but then COVID made it to the list. So now it's the 7th leading cause. It is also the 5th leading cause of death globally, and these numbers are based on the last week. I checked last week, and this website is actually daily, so you could actually check today and it probably will be more or less around the same ranking. But not only that, from my volunteering experience at Alzheimer's Disease Association and talking to many people who are suffering from early Alzheimer's disease or live with someone who suffers from early Alzheimer's disease, if there was one sentence that they all said in common, it would be something like, no one understands. And that's actually true, because Alzheimer's disease doesn't affect only an individual's physical health, but it also affects their emotional health, mental health, social life, and everyone around them. Last but not least, there is yet no cure to treat Alzheimer's disease. So what is Alzheimer's disease? Alzheimer's disease is an age-related neurodegenerative disease that is defined by neuropathological lesions in the brain that are important for cognitive functions. Over the years, many researchers have studied, of course, Alzheimer's disease to try to understand it more. And one of the well-known protein that has been associated or strongly associated with Alzheimer's disease Alzheimer's disease development and pathogenesis is called amyloid beta protein. First, let me introduce for you how this amyloid beta protein is being produced, because the rest of my talk I will be referring to amyloid beta a lot. Just briefly. So basically, amyloid beta protein, So amyloid beta protein is produced after a sequence of a cleavage of this larger protein that we have. Is called ABP, or amyloid precursor protein. Mainly two enzymes are responsible for this cleavage, which is beta secretase enzyme and gamma secretase enzymes. Basically, these enzymes cleave the ends of this protein, the N-terminal and the C-terminal. But what we really need to worry about here is the middle part here in purple. That gets released, and that's what is called amyloid beta protein. Now, the problem starts when this amyloid beta protein accumulates, when this process happens at higher rate than usual for some reason, or when there is some less efficiency in the clearing of this amyloid beta protein, so they end up accumulating more and more, and that's what has been linked to Alzheimer's disease development and pathogenesis. When talking about aging and age related disease, such as Alzheimer disease, there are so many factors that play a role in them, including genetic factors. Probably the first thing comes to your mind when I say genetic factors is a DNA. But here I'm talking about a molecular called circRNA. Let me explain for you what are circRNAs is. So the story starts when your mom and dad first met and decide to bring you to our beautiful world by passing their genes to you. So basically, you have half of your gene from your mom and half from your dad. And that is your DNA. Now your DNA go under the process called transcription, producing something called premature RNA. Let me tell you a little bit about RNAs, because I recently realized a lot of people just know protein and DNA and a protein, but nothing in the middle. So probably you heard about RNA in the recent days after COVID, because most of the COVID vaccines are RNA based, so people start reading more about RNA. So RNA is what we actually use to make proteins. And this premature RNAs, it contains many information in which some of them are important, presented in colored boxes here, and they are called exons. And some of them are less important, presented in gray lines here, and they are called introns. So this premature RNA goes under a process called alternative splicing. What is alternative splicing is really doing is cutting out this unimportant information and putting the important information back together. And it could do this in multiple actually combination, how they put the exons or the important information together, which then will be used to be translated into protein. And that's how we have multiple proteins or different proteins generated from the same premature RNA because of the alternative splicing. Now these proteins what actually do the function in our body. For example, we all know insulin is important to regulate blood sugar level, or collagen is important for strength of the skin, and so on. Now I want you to know that most of the time this is really what's happening with our premature RNA. But there are some times when this alternative splicing goes out of order or back spliced. That's what we call back splicing, resulting in the circulation of these premature RNA, and now they are called circRNAs. What's interesting about them, they do not translate to protein. So you end up having in your body something like proteins that doing the function and all these circRNAs that we don't know what they do yet. Let me go into more depth about what is back splicing and how they generate circRNAs. So during the back splicing, which I said is out of order or the other way around, a site of what's called accepting donor site, accepting site will join the donor site and form circulation. Now there are some introns, there are some sequence in these introns. They are called reverse complementary base pair matches. As the name suggested, they match each other. So they make the two ends come really close to each other, promoting the circulation to happen, as we could see from this figure. So when these circRNAs are generated or produced, they tend to have this unique sequence because they are backward. And these sequences are called junction snaps and reads, which we could use to detect the expression of these circRNAs, either by a strategy called real-time qPCR, where we actually look at the expression using out-phase primer, and then when the circulation happens, they meet, and then that's what we detect for the expression. Or we could also use a strategy called RNA-seq strategy sequencing and count the numbers of these reads, and based on that, compare it to the control and know if it's upregulated or downregulated, or upexpressed or down. Now, there is one challenge that we face when we're studying circRNAs. If you remember from the previous slide, I showed you that a lot of many circRNAs generate from the same premature RNA that also produce linear form of RNA. So in order to manipulate this or eliminate these, we want to do it in a way that we do not affect the linear form of the RNA, and therefore we don't affect also the protein associated with it, to know the function, what we actually think is due to the circRNAs. To overcome this challenge, we found that if we use a molecular scissor known as CRISPR and target these RCMs that are important for the circulation, we could actually inhibit the circulation from happening and therefore eliminate the circRNAs. Even though circRNAs is a very new field, many studies has highlighted some function of the circRNAs, and they want to go over them briefly. So circRNAs has been found to interact with many molecules, such as microRNA, by binding to it and inhibit its downstream targets or functions. It could also bind to RNA binding protein, influencing their downstream function, and that might also affect stuff like gene expression. circRNAs has been involved in DNA synthesis and ribosomal functions. So the main two questions that I wanted to ask during my PhD work is how these circRNAs are produced or are involved in the process of aging, and if they are involved in the aging, how they are involved in Alzheimer's disease development. To do so, of course, I could not do this in a human yet. So we use as an alternative C. elegans, nematode C. elegans worm, to do our studies in them. And that's for so multiple reasons. The first one, the C. elegans is the first animal with a complete genome sequence, meaning that we know everything about their genome, so we could actually follow what we found. It has a very rapid life cycle, 3 days, like from young to adult in just 3 days. They have short lifespan, meaning they live up to 20 days only, so we could do this aging experiment. Imagine if we do this in a human. To do Alzheimer, you will at least spend like 60 or 70 years in just one human, right? It's also very cheap, and it is available for genetic tools such as the CRISPR, and it also has complete map nerve system, so we also know everything about their nerves. And what is more important, it does share a lot of biological pathways with the human. So whatever we find in C. elegans, we might be able to apply it to a human for our own benefits. Now, to address our first question, how these circRNAs are involved in aging, we first looked at what has been known about circRNAs during aging. Many labs, including our own, found that circRNAs accumulate during aging in worms, flies, mice, and a human. And what I really mean by that is, as we get older, we have more of these circRNAs inside our body. There are multiple reasons why this could be the reason, but it's probably because of two features that circRNAs have, which is high stability and resistant to degradation. And that's simply because they are circular shaped. So they don't have this free ends make them accessible for degradation mechanism or pathways. So even though the circRNA expression is lower, way lower than linear RNA expression, but because they are high stable and resistant to degradation, they last longer in our body, and that's why they probably accumulate during aging. Now in worms, the model that I'm using for my study, we found that many circRNAs accumulate during aging. And what you're looking here at is RNA sequence data for all circRNAs, where each dot of this represents a circRNA. So anything above this red line represents a circRNA that has been abrogated or accumulated during aging significantly, and anything down from this blue line is significantly decreased. Anything in the middle is unchanged or insignificant. And the way we did this is by comparing animals at different time point or different age point. For example, we do day 1 adult animals compared to larvae 4 or larvae stage 4. Larvae stage 4 is basically 24 hours before the worms reach adulthood. And then after that, we call them day 1 adult, day 2 adult, and so on. So we did multiple comparison here, and we found, and you could also tell that globally, many of these circRNAs accumulate during aging. What is interesting from these circRNAs accumulating during aging is that some of them, they come from genes that has been linked to longevity or aging. So they are derived from genes that has been linked to aging. And from the list we have, we found that CRH1 is one of the most high abundance of age-accumulated circRNA, but not only that, we also know about the RCMs that are required for their mechanism, so they are simple to actually study than the others. So here we ask the question, what role do this age-accumulated circular CRH1 RNA plays in lifespan? We hypothesize that the expression of the circular RNA is detrimental to lifespan. And the way, the approach that we used to answer our question was by generating loss of function circular RNA CRH1 mutant, and then examine if there was any changes in their lifespan. Let me first tell you how did we generate this loss of function mutants. So remember I told you about the SPAC splicing? So CRH1 is actually, or CRH1 circular RNAs, they are produced from exon 4 of the host gene CRH1. And depending on where this splicing donor site bind, either splicing donor 2, splicing acceptor 1 or 2, we could, it could form two different circles, and the only difference between them are just really tiny difference, 6 nucleotides. So there are two circular RNAs that generated from the CRH1 gene. Now, we talked about the molecular scissor that we could use to inhibit the expression of these circles, which is CRISPR-Cas9 strategy. So we found that if we target these RCMs important for their circulation to happen, we were able to eliminate the expression of these circular RNAs. And we were able to do that in two different independent alleles. The only difference between them is the size of the deletion. But before we continue with our lifespan and see any changes in lifespan, we wanted to also again make sure we did not change the linear form of the CRH1 coming from the same gene, and therefore we did not also affect the protein associated with CRH1. To do that, we conducted a real-time qPCR where we actually, I don't know what's going on here, but where we actually looked at, I don't know what is that, but they are same words as these ones, sorry. So, but what we're actually looking here at is the fold change of expression of both circular RNAs and linear RNAs coming from the CRH1 gene. And we're doing this for different age time, during the aging. So here, for the both alleles, so both of these are worms of day 1 adults, and here worms of day 7 adults. And as you could still hear, both circles in all these four graphs that show expression of 0. So that means that we successfully eliminate the expression of the circular RNAs during aging. What is also really important here to notice is that the linear form, which is this one here in blue, in both of them, was unchanged. So we did not change the linear expression coming from the CRH1. And that's true at day 1 and day 7 for both alleles. We also confirmed that when we actually looked also at the RNA level using Northern blot, where you could actually also visualize the RNA level from a gel. And here what you're looking at is the mutant that we created compared to the wild type, just normal worms. As you could tell, for the linear, more or less, they're all the same. They all express the circular RNA, the RNAs in them. But now the circle one, it's completely gone from the mutant, and you could still see them in the wild type. I know some of you might think why we see them like really light like this, because I say circRNA expression is very low compared to the linear RNA in normally, but they are still expressed. To go further, we also wanted to make sure that protein associated with CRH1 is unchanged. And how we did that was by doing Western blot, where we looked at the protein level, or phosphorylated activated protein level of the CRH1, and compare it to the wild type, and also compare it to the linear mutant available. So here, the two alleles of circular CRH1 mutant that we made show similar level to wild type of the protein itself. So we did not change the protein. However, in the linear one, because they don't have that linear RNA important for the protein, in the mutant, they don't have the protein, so we compared to it and we made sure that, okay, so our mutation did not change the level of CRH1 protein in our alleles. So now we could actually go ahead and ask the question, what role do age-accumulated CRH1 circular RNA play in lifespan? Knowing whatever we see will be due to the circular RNAs themselves, but not to their protein or their RNA, right? So interestingly, what we found was that we found the mutation in circular CRH1 exhibit or extend the mean lifespan in worms. What I really mean, what we were really looking at here is what's called lifespan. So basically what we do is we're taking age-synchronized worms when they are adult, when they reach adulthood, and we count them every day, and we score whether they are dead or alive. Dead or alive. This is all I do in my lab. Dead or alive. Oh, okay? And then we convert these numbers into survival percentage over the time, and we look how long did they live. Now in the wild type here you see in the blue, just normal worms, this is their curve of survival, but if we delete the CRH1 circular RNA and them in two different alleles, or two different alleles, we found there was an extension of the mean lifespan. More or less, this extension was about 10% extension, more or less, depending on the allele. This is not too high, but imagine with me, we're deleting one circle of this in this worm, and we're already getting 10% extension of the mean lifespan. So that's impressive. So to conclude, or to summarize our first question, we found that circRNAs accumulate during aging in C. elegans, including CRH1 and many other circRNAs. In wild type, circRNA from CRH1 is detrimental for lifespan, because if we remove them, we get an extension of 10% or more. And by the way, this work has been published and available in Aging Cells, so if you want to go ahead and read more about it, there is so much interesting data that I did not cover today, so it's there. So now I want to move to my second question. Now I know this circRNA CRH1 plays a role in aging. So does it play any role in Alzheimer's disease development? So I hypothesize that the circular CRH1 expression is important for modulating amyloid beta-induced toxicity in Alzheimer's disease. In a second, I will explain for you what I really mean by amyloid beta-induced toxicity. But basically, the amyloid beta, remember, it was the protein that has been strongly associated with Alzheimer's disease that I showed you at the beginning of the talk. The approach we did was to examine changes in these amyloid beta-induced toxicity in the CRH1 circular RNA mutant that carrying or expressing a human version of amyloid beta inside them. To do so, we use a transgenic C. elegans worm that is expressing the amyloid beta, or a human version of amyloid beta, under the UNC54 promoter, which is a muscle promoter. So these worms express the amyloid beta in the muscles. And this amyloid beta, these worms expressing amyloid beta usually are just fine at lower temperature. And that's because the amyloid beta itself has not been induced. In order to induce the amyloid beta, we need higher temperature of 25 or more. Other than that, they are just normal. So what has been already reported that at higher temperature of 25, which now we have the amyloid beta being induced, these worms experience two things. The first one is that they live shorter, so they have shorter lifespan. And the second one, they experience severe paralysis, meaning that they cannot really move well. Here I will explain for you what I really mean by paralysis, or what I'm really looking at. So I'm looking at the full body movement of a worm after I touch it with a wire. So basically similar to lifespan, I'm having this synchronized worm when they are adult. I put them, I introduce them to 25 degrees to have this amyloid beta being inducted, and then count every day. But this time I'm really saying, is it paralyzed? Is it not? Is it paralyzed? Is it not? So basically, or in normal situation, when a worm is, when I touch a worm and it's not paralyzed, it can just move around completely like that, like you see in this video here. But if the worms were paralyzed, when the worms are paralyzed, they fail to move their body. You could see their head still moving, but they fail to have full body movement. And that's what really I count, okay, this is paralyzed, this is not paralyzed, over the days as well, during aging. So I asked the question again, do CRH1 circRNA play a role in this amyloid beta induced toxicity? And that's what I mean by amyloid beta induced toxicity, lifespan and paralysis, okay? To do so, first I wanted to confirm what was already reported about this worms expressing amyloid beta in muscles that actually do have shorter lifespan, and they do also experience severe paralysis at higher temperature. So I was able to confirm that in these two figures. In the right side here, yeah, my right side, your left, I guess. You see in the black, this is the worms that expressing amyloid beta in the muscles, and at higher temperature, which is after amyloid beta induction, they live shorter, or they have shorter mean lifespan compared to the wild type, just normal worms that they do not have amyloid beta. Similarly here in the paralysis assay, they experience paralysis at 25 degrees, but the wild type worms, they do not experience paralysis at all, which makes sense because they don't even have amyloid beta in them. Now, if I remove that one circular CRH1 from them, you could see in purple here that they were able to restore this lifespan of wild type. So the only difference they have from this transgenic worms is that now they have a mutation in the circular CRH1 mutant, meaning that they don't have this circular CRH1 in them. And similar results were also found when we did the paralysis assay. They were able to improve or reduce this paralysis effect that the worms experience after we remove the CRH1. And we did this in two different alleles. Of course, you could see it didn't go back to normal, but it did do a lot of the improvement. So we conclude with that genetic removal of CRH1 circular RNA provide protection against amyloid beta toxicity. But the question is, or let me first put things together. So in the transgenic worms that expressing amyloid beta in the muscles, if you remove CRH1, just remove one CRH1, okay? And after amyloid beta induction, which happens at higher temperature, the worms benefit by two things. They have longer mean lifespan now, and they reduce paralysis effect. Now the question in wild type, if they just transgenic worms that has amyloid beta in them, and normal level of CRH1, what really happens after amyloid beta induction? Does it change the expression of CRH1 circular RNA during aging? So again, here we're looking back at real-time qPCR, we're looking at the fold change expression of the circRNA and the linear RNA from the CRH1, and comparing during aging if the amyloid beta induction at higher temperature did increase the expression of CRH1. So first of all, in wild type here, and by the way, we are normalizing to day 1 adults, so different time points. So in wild type here, we did confirm that CRH1 does increase during aging, even at 25 degrees, when we compare it to day 1 within the same wild type genotype. And that's also similar in the transgenic worms that carrying amyloid beta. At 25 degrees, they, for adult worms, has more or increased expression of CRH1 circRNA compared to day 1 at 25 degrees. But what is interesting here is that the ratio of the increase in the transgenic worms carrying amyloid beta is even more now, more by like 1.2. So we did similar thing, but this time we decided to look just at the transgenic worms with the same age, so both of them are at the same age, day 4. We can compare them to each other, but this time the only difference is the temperature. So 25 after amyloid beta induction, 20 degrees before amyloid beta induction, to see if this amyloid beta induction did anything to the expression of this circRNA. And indeed, it did actually increase one of these circRNAs significantly. It did not do anything to the second part of the second RNA coming from the CRH1. And it did also upregulate a little bit the linear one as well. So now we ask the question, does loss of function of circRNA expression reduce amyloid beta aggregates in the transgenic worms expressing amyloid beta in the muscles? And what I really mean by that, remember this figure I showed you at the beginning? Remember I told you that amyloid beta accumulation is the problem, and that is associated with Alzheimer's disease. So that's what I mean by aggregates, amyloid beta aggregates. We actually could even visualize these aggregates and count them and see if they are more or less, which I will show you in a second how we did that. But before to look at the amyloid beta aggregates, we wanted to make sure that the loss of function of CRH1 mutant did not change the amyloid beta mRNA level itself. So it didn't somehow change the expression of this, because that would make sense if the loss of function CRH1, it changed the expression of amyloid beta mRNA level. So that really will make sense why we have more amyloid beta aggregates. But we wanted to make sure this level is not changed, so they are at the same level during aging, but the only thing is changing is the amyloid beta aggregates. So again, we did RT-qPCR to look at the relative quantity of the mRNA level of amyloid beta during aging, day 1, day 2, day 3, after amyloid beta induction at 25 degrees. And we found that there was no significant change during aging in both transgenic worms that expressing amyloid beta in the muscles, and even after we delete the CRH1 in the expression. So now we could actually go back and look at the amyloid beta aggregates and count them and visualize them using a staining called X34 staining. What is this staining is doing? It's in vivo staining, so we could actually use to visualize this amyloid beta aggregates or accumulation in a living animal, and they bind specifically to the amyloid beta. So it will bind to amyloid beta, and under the microscope we will be able to see it and count them. I'm going to show you an example. Here you are looking at the head region of the worms of 3 different genotypes. This one here is a wild type that does not have amyloid beta, just wild type, completely wild type worms. And then the one in the middle is the transgenic worms that expressing amyloid beta in the muscles. And the one in the left is transgenic worms expressing amyloid beta in the muscles, but this time they also do not have CRH1 circRNAs in them, right? And the amyloid beta aggregates are shown as puncta here. If you could see from the white arrow, they show as a puncta here. And this is how we could visualize them. As you could tell, in the wild type, we don't see a bunch of these puncta or amyloid beta aggregates, which makes sense because they don't even have amyloid beta in them, so they don't have this aggregation in them. In the transgenic worms, we see that they have a lot of these puncta, but after we remove the CRH1 circRNA expression, these puncta become way less. Thomas, a former undergrad in our lab, Sandra just mentioned in the beginning, he was able to quantify the number of these puncta of amyloid beta aggregates using MSJ software. And he confirmed to us that indeed, after CRH1 removal, CRH1 circRNA removal in the transgenic worms, the number of these amyloid beta puncta significantly decreased. Again, it did not come back to wild type because they still suffer from paralysis, but not as severe as the transgenic worms. So I'm gonna put back things together. So in a transgenic worm, if we remove that CRH1 circRNA expression, the worm will benefit by longer mean lifespan, reduced paralysis effect, and now reduced amyloid beta aggregates. So we ask, what is it in the CRH1 circular mutant that is different and might be responsible for all these changes that we see, right? Previously, we did show that in CRH1 circRNA mutant, many genes has been upregulated and many genes seems to be downregulated compared to the wild type. So here you're looking at the RNA sequence data where each dot represents a gene, and anything above this red line is significantly upregulated genes, and anything below this line is downregulated, significantly downregulated genes. Anything in the middle are unchanged or non-significant. Interestingly, among upregulated genes, we found that a subgroup of them were collagen encoding genes. And the reason why we wanted to look or focus on collagen, because collagen indeed has been linked to Alzheimer's disease, but not only Alzheimer's disease, it actually has been linked to amyloid beta formation, clearance, and so on. And these are some papers if you guys want to check at some point, and there are many studies that suggested collagen is linked to amyloid beta formation and clearance. So we ask, is it possible that collagen is responsible to the phenotype change that we see in our case or in our scenario? So from 21 upregulated collagen encoding genes, we decide to focus on 6 of them based on their previous knowledge or previous link to aging, especially COL49 and COL179. They are strongly, has been reported to be linked to aging. So we wanted to focus on them, and then we tested whether these collagens are up or down regulated in the CRH1 circRNA mutant carrying amyloid beta expression in the muscles compared to the transgenic worms where just amyloid beta expressed. At day 3 adults, I choose this day because they are more or less paralyzed at 25 degrees after amyloid beta induction. And we found that out of these 6, only the collagen 49 shown to be very upregulated. Unfortunately, there was no available null mutant strain, so we had to make CRISPR-Cas9 or a mutation mutant using CRISPR-Cas9, the molecular scissor that I told you about. And we were able to delete these exons important for the protein of COL49 and we made this a mutant. It's a mutation in COL49. Then after we had this mutant, we genetically crossed these worms with the amyloid beta transgenic worms. And guess what we're gonna do next? We're gonna look at their paralysis, right? So we did look at their paralysis. As you might expect it already, that they now, after removing this COL49 from them, after removing the COL49 expression from them, they experience even worse paralysis. So, as a control, these worms by themselves, COL49 mutant without the transgenic gene, they experience no paralysis at all, shown here in red. But if they have amyloid beta in them, they experience very severe paralysis, worse than the transgenic, the wild type of COL49 transgenic genes. So that leaves us with a question: how does the circRNA modulate COL49 gene expression? To answer this, we need some future experiment. To do so, including what's known as circRNA RNA binding protein roll down, where we actually could use some probe hybridization and pull down any proteins that bind to CRH1 circRNA, and then after mass spectrum, we could identify these proteins and see if they have been previously reported to be involved in the COL49 expression pathways. We hypothesize, or we propose, that the model is that CRH1 sequestered the RNA binding proteins that are important for COL49 messenger RNA expression. Basically, in wild type, when you have this circular CRH1, it will bind these proteins, RNA binding proteins, and maybe act as sponges somehow, we don't know how, leading to just a normal level of expression of COL49. But in the mutant, when you don't have this circular CRH1, maybe more of these RNA binding protein are now available because they don't bind to it, and somehow they lead to higher expression of the COL49 expression. And of course, more research need to be done, or experiment need to be done to answer these questions. So that brings me to my summary slides, or take-home messages, that C. elegans is the best, is a very good model to study aging, especially in regard to circRNAs. Many circRNAs, including CRH1 and many others, increase during aging in C. elegans, also in many organisms, and that we could use the CRISPR editing, gene editing, to target the RCMs that are important for the back splicing or the formation of circRNAs, to be able to manipulate circRNAs, and that in wild type CRH1 circRNA expression is detrimental for lifespan. Also, the loss of function of circRNA expression improves amyloid beta phenotypes, which means paralysis and lifespan. And any work that I mentioned in relate to amyloid beta or Alzheimer is now available at bioRxiv if you want to read more about it, and soon will be submitted to a journal. What else? So, of course, I started my talk talking about a human, and all of a sudden I'm talking about this tiny worm, saying it's the best model, but you're probably wondering how these findings are benefit us human, right? So I said, in C. elegans, if you delete one circular RNA, the worms will benefit by longer mean lifespan, delayed progression of amyloid beta induced toxicity. So imagine if that was also true in a human. We don't know that, but we also know that C. elegans in a human does share a lot of biological pathways. So imagine if that was true in a human, then these circular RNAs could be used as biomarker for early detection for Alzheimer's disease, and or therapeutic target to treat Alzheimer's disease. With that, I want to acknowledge and thank some people. First, current and former lab member in our lab, starting from Dr. Alexander van der Linden for all the support and coffee, that free coffee that he gave, and then everyone else in the lab. I also want to thank members, Dr. Pedro and Dr. David Knob, because they collaborate with us in the aging cell paper. Our funding, our community members, Dr. Tom, Dr. Grant, Dr. May, and Dr. Simone, which they probably never know that I select each one for a reason, because you made me change my major, Dr. May, and both of you made me be in C. elegans lab, which I really wanted to be in C. elegans lab, and Dr. Simon gave me a lot of how to find a good lab. I also have a special thank to Dr. Monica, because she was the first one to give me opportunity to do internship in her lab to work with C. elegans, so she was the reason why I really like to work with C. elegans. Dr. Saito, because he helped me during this internship, and he was the one inviting me in Japan actually, so he's following up with me all the time. My family and my friend, and my forever emotional support, my dog Sugar. And now I could take questions, but if you allow me, please, I see like some Arabic speaker here. They came to support me, so as an appreciation, I want to have this slide translated in Arabic just up there for them. If you guys have any question, ask me, but I could start taking questions from you guys.